Thank you, Dr. Greenwood. Howdy? Howdy. Come on. Howdy. Howdy. Geez, all right, good deal. Well, it's great to have you guys all here this afternoon. Thank you for being here. I didn't mean to be mysterious about the topic, but I didn't want to do another science talk. You guys are going to hear lots of science talks, and I'm going to work some science in. But I wanted to reflect. I was reflecting back on when I was where you were. I had a lot of questions as to where I was going, and so I want to give a presentation and give you a little bit of an idea of where I've come through, because I hope during that process you can get some senses, maybe some ideas that you can take forward. One of the things, and I've got this kind of weird, this is a slide of the University of Tennessee at Knoxville. That's where I did my doctorate. And when I arrived there, I went to a very small high school, 12 students in my graduating class. I did my master's degree, my master's degree and my bachelor's degree at a place called Northeast Louisiana University, which is the University of Louisiana Monroe. And so when I got to Tennessee, I really had not been exposed to big science. I'd done thesis and some other things. And so I can remember being at a place like this, like A&M, really wondering what the heck I was going to do in the future and not knowing. And so I want to share some of that with you and some of my thought process with you about that. But this is going to be a little bit different presentation in that I'm trying a different technology. Some of you have seen this. This is kind of a clicker-based kind of thing, but you pull out your phones, or if you've got your you got your computer, you can either go to your phone and text Tim Lightfoot 930 to the number 22333 to join. There's going to be several questions today, and you might win prizes. Or you can go to a website. Actually, this is probably the easier way, is go to a website, go to pollev.com/timlightfoot930. And when you get there, when you get there and get in, I want you to answer the question, I'm here today at this talk because I have to be. It's a class I have to take. The topic sounded interesting, and I have nothing better to do, or I have nothing better to do on a Monday afternoon. So you should be able to get in one of those two ways. And whatever way you get in, that's the way you're going to stay with it during the rest of the talk today.
So I'll be privy to who's answering what relative to their grade?
No. Sorry.
It's a requirement, so that's that.
But it's not surprising. This doesn't surprise me at all. I kind of anticipated. But really, the purpose of this slide is to get you guys into the system so you can see it and get familiar with it, because there's going to be a couple other slides coming up where you actually have to answer some things. Okay, everybody responded that's going to respond at this point? Excellent. Well, since you are here because you have to be here, hopefully by the end of the class you've actually learned something out of all of this. And so one of the things that I want you to learn is that you will never succeed unless you do it with a group of people in life, period. I get a lot of credit for a lot of things that a lot of other people do to help me push things along. And I wanted to share you, if this is about a community that you're involved in, and I've been fortunate to be involved in this thing called exercise physiology for a long period of time. This is my current lab group. This is last year's picture. Actually, we're taking this year's picture at 4 o'clock today. But we have an undergraduate on the left, that's Chaz, and we have 3 doctoral students. We have Jorge, we have Aylin, and Kat, and if you don't know them, they're around. We've got several other undergraduates involved as well. It's about the colleagues that I have around the country that I work with on a continuum basis. Here's a list of their names at a variety of different places. It's also about you as taxpayers, because you all have paid for some of the work that we've done that you're going to see today. So we've been supported by the National Institutes of Health. We've been supported by the Department of Defense. So it takes community. And on top of that, I want to share with you this slide. This is my former students that have been with me on this journey since I started as a faculty member in 1989. These are undergraduates primarily, but there's some master's students in there and a bunch of doctoral students as well. And so my first point for you is that as you look around the room and you make friends with the people in this classroom and you work in labs and you meet faculty, these are the people that are going to help you through the rest of your career, that you're going to stay with. And so don't ever forget all the people that you work with. Now today I'm going to do a little bit of introduction, which you've already got. We're going to talk about some of my early forays into blood pressure. I worked with the space program starting in about 1984, and I'll tell you a little bit more about that in a few minutes. I'm going to talk to you about how we moved over to do genetic stuff, which is what we have done since about 1999 to the current time frame, and then some implications, which I think have some big implications for us, how I think the future is coming, and to give you some take-home messages. Okay? So the first thing I want to do is give you a little bit of introduction. This is what I looked like when I arrived at the University of Tennessee in 1983. I was a little bit clueless, as most people are when they finish a master's degree and they think they know everything in the world, and what they don't realize is what they don't know. But I was really fortunate that I got to work with some really great people. This first guy here is a guy named Kelly Stewart. Kelly died actually when he was 34. He was a faculty member of mine in my master's program, who is the first one that ever looked at me and said, "You ever think about being a faculty member, a professor?" That was something that never entered my mind. And so let me ask you all that, or tell you all that. If you haven't thought about it, you should think about it. It's certainly a job career path that's available to you. Through Kelly, I wound up getting into the University of Tennessee, where I wound up working with my advisors, a guy named Dr. Ed Halley. I also worked with a guy named Don Franks, who always wore the ugliest clothes ever. And he did that on purpose to distract you because he's a brilliant kind of guy. And then this guy, Hugh Welch. And these were wonderful mentors. Come on in, Jorge. These were wonderful mentors and have turned out to be lifelong friends of mine. And again, this comes back to this idea about this is a community that you have joined, that you will work with people throughout your career. One of the things when I got to Tennessee, Dr. Halley asked me, "What are you interested in working in?" And I said, "I really don't know. I'm really kind of excited about space physiology." And he said, "Well, I don't know anything about that." Well, let's find somebody. And so we happened to go to Southeast ACSM meeting when there was a lady there named Diane Spittler, who was in charge of the Aerospace Physiology Lab at Kennedy Space Center. And she was doing a poster, and Dr. Franks in particular said, "Go talk to her. I know her. I'll introduce you." And I'm still kind of a shy guy, but he said, "Don't be shy. Come and talk to her." And so I went to talk to Dr. Spittler. She invited me to come to Kennedy Space Center, where they were still launching rockets and people, and they were working with the astronauts. And she said, "Come down and spend 2 weeks and see what's going on." So I did. Paid for my ticket, took a bus down, lived in someone's house for 2 weeks, and it was a great time. Unfortunately, Dr. Spittler actually had passed away with brain cancer between the time I'd made the arrangements and the time I got there, so I wound up working with this lady, Dr. Marianne Fry. Dr. Fry was great. Really opened a lot of doors for me in the space program. And I wound up going back there for another 9 months the next year to do my dissertation work there. And so at the time, NASA was very concerned about the effects of spaceflight on the regulation of blood pressure. It appeared that athletes actually had compromised blood pressure control. And so if you sent them up in space and then you brought them back, they actually had a tendency to pass out. And I don't know if you've ever noticed, if you looked at the footage of the Russian astronauts, when they bring the Russians back, they actually carry them out on a litter. And it's not because that's the way they do it, it's because they can't stand up without passing out. So NASA was very concerned about how aerobic training in particular, but also resistance training would affect whether or not someone could actually stand upright for a period of time. Now during this time, I wound up meeting a lot of people when I was at NASA. Here's a picture of us at a launch. When they did launches, they would let us go out. Actually, this is the vehicle assembly building, which is where they used to put together the big Saturn V rockets. That's me right there in the middle on top of the van, but this was the whole staff of the biomedical crew, and the ones that weren't up over in the launch control facility, which you can't see right there, kind of through the, we all congregated there and watched the launches. These were wonderful people, and this comes back to my idea about this is a community that you wind up getting exposed to. When I went to Tennessee, I had no clue that within 2 years I would be at NASA working with the astronauts, working with these people to shoot them up in space. So the second point here I want you to make is follow your passion. Whatever your passion is. What really jazzes you up right now about what you're doing? And so we're going to come back to, this is our next poll question. So for those of you that have joined the poll, if you haven't, Jorge and this gentleman in the back that just came in, go to this site either on the, you can either text it or go to the web, and put in what are you most interested in professionally right now? What are you most interested in professionally? Like that. Now, these are great responses. And the bigger they are, the more people are texting that in or putting that in. But you can start to see, for a challenge for any speaker for this group, is this is a wide range of audience. And so what I want to really, again, encourage you, figure out what your passion is and go for it. Even sitting in seminars like this, and I'm going to bring a story like this up in a little bit, you never know what you're going to pick up. And so this is great to see. This will help Mike in particular when he thinks about speakers for this seminar a little bit later on. We see a lot of interest in sports performance here, which is really cool kind of thing. Let me give you a little bit more of the story about NASA. What I learned to do at NASA was how to use a vacuum cleaner to do science. Okay? So we had this thing called, we used a chamber called a lower body negative pressure chamber, or LBP for short. And here's a really small picture. I'll show you a bigger one here in a second. But here's the idea. What you do is you lay somebody down in this chamber, you put a spray skirt on them here at the waist, you close it up, you put a big vacuum cleaner on this, and you pull pressure out of the chamber. And what it does, since it reduces air pressure in the chamber, your body then pools fluid, primarily blood, in the lower part of your body. And so everything, all your blood pressure control mechanisms in your brain and in your heart suddenly think that you're losing volume, that you're bleeding. And so you start to see all the same things that would happen as if someone were actually bleeding. And you can turn this off, and when you turn it off, immediately, of course, they come back. We could also take a vacuum cleaner and put on a chamber on people's neck. Your primary blood pressure controllers are in your neck. And so what we can do with either pressure or with vacuum, we can actually either extend or compress those receptors, which can control your heart rate and control your blood pressure. So we could do a lot by using vacuum cleaners. We could look at whether or not someone was going to be able to stand upright or pass out or not after training. And so that's what we did a lot of with this. This is a picture of the NASA LBP box I worked with. This was a plywood box. This was one they actually started using this in 1968 on the Apollo astronauts when they came back. This guy right here is Don Doerr. He was, if you see all the movies about the Mission Control Center, he was the biomedical engineer on the board for NASA. We had many interesting talks. He predicted the space shuttle Challenger would blow up before it did, but that's another story. Here's the LBP box that we made in 1986. This was a plexiglass one, and I carried this box around for 30 years doing a variety of different things with it. It's now in Michigan with one of my former doc students who's using it on race car drivers, looking at G tolerance in race car drivers. So there's a lot of different things that you can do once you figure out what your passion is, because I never thought that I was interested in space physiology that this kind of stuff would come out of it. So just some questions that we answered. What NASA was very interested in, would endurance training cause people to pass out quicker? The answer was no. What we did find was that acute resistance training actually increased their tolerance. So actually, if you want to make astronauts more tolerant to space, what you do is, or fighter pilots for that matter, you do resistance training with them. We also found that if we actually put people in this box over and over again for 10 days, we actually increased their tolerance by 55%. And so we actually provided a paradigm for the Air Force. The Air Force was very concerned about their fighter pilots. So this was a paradigm that we actually provided for the Air Force so they could increase G tolerance in their fighter pilots as well. But you know, it's interesting. I had to go to a seminar when I was at Johns Hopkins. I did a postdoc at Johns Hopkins. And I was at a seminar. We had to do a seminar every Thursday afternoon. It was called the beer seminar because there was beer. And but they actually put a limit on it. I know. Yeah. Actually, Texas A&M doesn't allow alcohol in classroom buildings. I know. But we actually have a seminar right now we call beer seminar. It's in a non-university building, but we'll leave it alone.
Some of them come in here drunk, so they don't really.
No, they don't. All are awake and with me today. But you know, I was at this seminar in 1989. It's one of these seminars that I went to every week. And I happened to have a friend of mine, a guy named Dr. Steve Kleberger, who was presenting at that time. And Steve had been a subject of ours and so forth. But he was into genetics. That was what he did. And he gave a seminar on a new model that they were using. This was in 1989 on a new mouse model that they were using. He was looking at lung respiratory sensitivity. And I'm not interested in lungs and respiratory sensitivity. That's the worst part of exercise physiology, is my opinion. But I sat there. But what struck me as I went through the whole session was that what Steve was telling us is that we could figure out where in the genome was controlling any characteristic. That was a model that they were using. Where in the genome that you could control any characteristic? And I asked Steve, I said, can we apply this to exercise? He said, as long as you have a stable characteristic, absolutely. And I thought, wait a minute, this is really cool. I'd been interested in genetics since I was a sophomore in high school, kind of one of those geeky kind of athlete kind of guys. And it was like, wait a minute, we can do genetics of exercise. This is really cool. And no one was really doing anything at the time. And so I thought, I can measure VO2. That's a pretty stable characteristic in humans, for sure. Why not in animals? I can measure it in humans. Certainly I can measure this in mice. And so this was our first attempt at a VO2 measuring treadmill. I think in 1991 was when I built this. The treadmills at the time for mice cost about $26,000, and we didn't have $26,000. So this was 3 tomato paste cans, big tomato paste cans, and all lined up, and this is a belt sander back here. We used a belt sander because we could run that DC so it wouldn't speed up and slow down. We built this plexiglass chamber. You put a mouse right in the top, and it was all connected to the VO2 cart back here. And that's another side of it. That a little hole we put in there with a finger on a rubber glove so we could actually tap the mouse's tail so they would actually run, because they don't like to run. Mice don't like to run. And we had actually I'll show you on this one. We had maybe not. So we had problems with that. That didn't work very well. And we were hearing from other people that treadmill running and VO2 measurement in mice wasn't working very well. So we said, well, maybe swimming is easier. So this is one of my master students, Russ Gilbert is his name, and he built a swim flue. So this is a little chamber that we could put down in the water, and we could put the mouse in there, and this is all hooked up to a metabolic cart here, and we did all of our data analysis. This is a little spirometer that Russ built for the mouse. So we were trying to measure VO2 in mice swimming. It didn't work too well. Mice, number one, don't like to swim. They go to the bottom and then they pop up, and they go to the bottom and they pop up. And you wound up getting water and everything, so that didn't work out too well. And so this one never got out of beta stage. We tried this again. So we were still doing all the blood pressure stuff, but this was something that we were interested in. And so my next take-home message for you is that never give up on something that you're interested in doing. Now the first date was in 1991. We really didn't start doing genetic stuff until 1999. And so it took us about 8 years to get to the next stage. And part of the reason we got to the next stage was because we went to Southeast ACSM and we heard this guy talk, Steve Britton. He's at Michigan. He and his colleague Lauren Koch. And what Steve suggested, he was having real problems with mice on treadmills. And so what he suggested, why don't you just test their endurance instead of their aerobic capacity? Just put them on a treadmill and see how long they go before they stop running. Just go, wow, that seemed to be, I came from a human mindset when we could all measure VO2. He said, why not just measure how long they run? Oh, okay, we'll try that. And guess what? That's what everybody now does. Because no one's been able to measure VO2 in mice. And I'm not going to give you the whole story, but we have continued to try to measure VO2 in mice. And for a variety of reasons, we can't. And I've spent probably $100,000 trying to do that. So, yeah, it takes a while, but I learn after a while. So, why don't we test endurance instead? And so when we did this, here was the first time we did this. We took a regular treadmill, a cardiac treadmill, as a matter of fact, that would run slow enough, and we built this grid on it, and that's a shock grid in there, where we could put, in this case, 5 mice in there. This is actually the second grid we built. The first grid we built was plexiglass, and it looked really cool, but the walls were too close together, and the mice learned very quickly they could take it all up on one wall and one on the other, and they do that with their back legs as well, and they look at you through the plexiglass. And at that point you go, wait a minute, a 20-gram mouse is outsmarting me. No way. So then we put Vaseline on the wall so they couldn't do that. And I don't know if you've ever tried to catch mice that are greased in Vaseline, but it's not easy. So these are the fun things that you get to do in the lab when you're trying this stuff. So anyway, so this was actually our second attempt. That's why it's not very pretty. I wasn't going to spend a lot of time building something really pretty if it didn't work. And so we put it on top of a treadmill that would run at the speeds we needed, which was very, very slow speeds for humans, but really fast for mice. This treadmill was an old treadmill that had been retired because it wouldn't elevate. And we realized then we needed it to elevate, so we had another treadmill that would elevate that wouldn't run. And so what we did, we put that treadmill on top of the treadmill that would elevate. And you could see that everything then worked fine. So, you know, you do what you have to do sometimes to make it work to collect initial data. So we had continued, again, so this was our mindset. I was changing my mind about what I wanted to do in research. I was really interested in the genetics thing. We had continued to do some LBMP stuff. We couldn't make the case. I was getting bored and frustrated. And so this change in measuring endurance really erased the barriers for us to do the endurance work. And this picture is actually a good friend of mine, a guy named David Bassett, who's the department chair at the University of Tennessee now. In his earlier Wildman days, we were actually measuring blood volume in humans, plasma volume. Actually, you inject a dye, and then you take a sample a little bit later on and do all the analysis, and you can measure it. It was a technique from the 1940s that we found that's very non-invasive. Only one company in the world makes the dye that you can use, though. So interesting things. So in 1998-99, we went full-time mouse work. So here's the next question on the poll that I want you to answer. What has been the biggest change that you have made in your professional life to this point? What's been the biggest change that you've made in your professional life? Because what I have just told you about is a change that I made in my professional life. I changed my topic completely. What's been the biggest change that you've made? That's a big change. Any others? I see someone still working on one. So I salute all of you. These are big deals. These are big deals to every one of you. And I mean, as you look at this list, some of these things are really difficult. I mean, all these things are very difficult to do. And so I really give you kudos for doing that. And I'm so pleased that you're here and that you're focusing on this. What I would encourage you is that the nice thing is you're in a business that allows you opportunities. And so even after you get your degree, you may find that you may wind up changing your focus again. Or you'll make other professional decisions. So what I would say is please don't ever be afraid to make that change. If your gut and your passion is telling you need to do something, you need to do that stuff. Again, I got to a point where I was bored with what I was doing. God, I can't tell you how how many times I stood beside that LBP box and listened to the vacuum cleaner go on and go off, and you watch, it's great, okay? But after a while, it's like, you can only do that so much. So making a change like this is a big deal. And again, I'm proud of all of you who have done that. So that when we made that change, I had an undergraduate student that first started doing our first mouse stuff. And when we published that, this was our first graph, our first mouse graph. And this was 2001, which was only 16 years ago. But this was the first time that anybody had taken a bunch of different kinds of mice and seen what their exercise endurance was. Actually, this was I want to say it was the first proof that there was genetic effect on exercise endurance. And so what we were measuring here was how long they would run. You put them on a standard treadmill protocol, how long they would run. And these are specific types of mice that are basically genetically all the same. So these are what they call inbred strains of mice. And so brother and sister, mother, dad, they all have the same genome. They are all exactly the same. So it makes it easier for us to do genetics work. And so what we were seeing was, in this case, that there were differences which indicated that there was some genetic variability here, and some genetics was playing some role in this. So I was presenting this at a conference in Portland, Oregon, and this guy walked up to me, Ted Garland. Ted is now at the University of California, Riverside, and he asked me a simple question. Ted said, would they do the same, would they act the same if you just put a running wheel in their cage, instead of forcing them to run on a treadmill? And I said, I don't know. Ted asked that because he was doing this kind of stuff. He had actually, since 1993, had been breeding mice to be high active. So he would take a litter of animals, and the high active ones, he pulled them out and he bred them together, and the next generation, to let you know, he's now at generation 93. He's continued to do that since 1993. But that question from Ted actually set us off to a little bit different direction. And instead of looking at exercise endurance, we said, wait a minute, is there any genetic control of how much activity we do? And we've often said, we call it, can you be born a couch potato or not? And so my lesson here is, don't hesitate to talk to strangers. You're not children anymore. You can talk to strangers. Ted's kind of strange at times. Let me tell you, I've continued to work with Ted. We're working on a paper together right now. He was here in March. And Ted can be a little bit strange, but don't ever hesitate to talk to strangers, because they got some really good ideas. Because at that point I thought, wait a minute, it's more important for us to figure out if people have a genetic predisposition to be active versus whether or not they have endurance. It's a bigger health question. So then we started looking at mice on running wheels. You might ask yourself, is this a good model of human voluntary activity? Because that's what we're trying to figure out, and I'll give you a little bit of data on that. You see similar responses in humans, and mice in particular, in cardiovascular functioning, in muscle and mitochondrial enzyme activities, and brain neurotransmitters in particular. If you put mice on a treadmill or rats on a treadmill, actually the brain neurotransmitters go the opposite direction than humans do, because it's a very stressful situation for rodents. Whereas if you give them a wheel and let them run, you have very similar responses as you do with humans. That's kind of sold it for us. As a matter of fact, what's also interesting is that if you put a mouse on a running wheel, they will run at about 70% of their max intensity. And if you give a human a treadmill and say, run just as fast, however fast you want to run, they'll run at about 70% of their max capacity. No matter if they're a diseased population or what, it's called self-selection of intensity. So it's about 70% in all the different species. So we thought, okay, this running wheel thing may work pretty well. And so we then asked this kind of question of, is there this genetics of physical activity? There was a guy that published a paper in '98 named Tom Rowland. It's the only paper he's ever published in this area. And he suggested, he did a nice review of all the literature at the time. He said, yep, absolutely, there can be the genetics of activity. And so our question then became, can you be born a couch potato? Can you be born to be predisposed to be inactive? Or can you be what we call a frantic banana? We didn't know what to call something opposite a couch potato. This was actually my wife's deal. She said, why don't you call it a frantic banana? I thought, oh, okay, that's pretty cool. We'll do that. Now, many people will say, no, I control how active I am. It's not my genetics. But I ask you, I challenge you, think about people in your life. Do you know people who are just refuse to get up off the couch? Do you know people who can't sit still? Absolutely. And so there is some indication that, yes, you can be born a couch potato or you can be born a frantic banana. And that's really irritating after a while, isn't it? So we did a lot of experiments. We've actually we have looked at, I think, 43 different strains of mice. And we found our high active strain of mice, our frantic bananas, is a strain of mouse called a C57LJ. And just to give you a sense of how far C57LJs run, if you take, and you can't see it very well, but this is Google Map, and College Station is right here. Here's Houston down here. Here's Temple, Waco, so forth. If you just let this mouse loose and let it run for 3 weeks, it will run almost 140 miles away. That's a high active mouse. They run anywhere from 6 to 10 miles a day. This is a 20-gram mouse, 20-gram mouse. Scale that up to humans, and that's a human running over 100 miles a day. Amazingly active. Okay? On the other hand, we also look for our couch potato mice, which is what we call which is called a C3H, HEJ. And if you compare them, do the same comparison, after 3 weeks, they'll get about 15 miles out of town. That's about as far as they go. And so these two strains of mice have formed the basis for a lot of our work. We've done crossbreeding. We found that high activity actually breeds through to the grandchildren, and a variety of other things. So we have been able to answer some questions. Can you be born a couch potato? The answer is overwhelmingly yes, and it's not just us. I think, so I'm going to give you this list. There are 15 articles here, including I think 5 from our lab, a variety of others. There's a bunch of human. Here's a bunch of human ones. Look at, here's one, this one, Janine Stubb. I think it's this one. Yeah, she got 2 of them. This one is from 87,000 people in it. The Europeans have a big twin database, and they've published a lot of this stuff. And so this is the heritability. So this is how much, what percentage of your activity is due to genetics. That's 85%. So you get a sense that it's very heritable. As a matter of fact, this is only 15 studies. There are a total of 47 studies out there in the last 20 years that have shown that your daily physical activity level is heritable. Okay? So it's not people talking about sidewalks, whether or not you have sidewalks and you're safe, and that's absolutely important. But a majority of whether or not you're active or not is what is your genetic predisposition. Okay? So here's our next question. Do you think you can be born a couch potato or a frantic banana? Easy. Yes or no? And maybe you're going against some of your training. It went against my training. My training was that we have complete control over that. Good, we haven't convinced somebody. All right, so the majority, we can do statistics on this. So the majority of the folks in the room think that you can be born a couch potato. And from what our data was showing and from what the literature has shown, that's what we think as well. And so the next question then becomes, if that's the case, then what are the genes that control this? What are the mechanisms? And we've really spent probably the last 10 years trying to figure that out. And so here's the map. I'm going to just show you some of the highlights of this. This is the map. This is a genomic map, and this is the mouse genome. There have been at least 6 studies, and they're listed here. These are 6 studies that have actually identified places in the genome that appear to be controlling activity. Now, there have been 2 human studies that have done this. There's only been 2 human studies. They've been very small. But I took and transformed those positions, those genomic locations, to the mouse, since the majority of this stuff has been done in mice. And so what you can see is that this part of chromosome 1 seems to be associated with whether or not with exercise participation. Here's duration, whether or not inactive or not, distance, duration, how far they run, how long they run. And so what you can do is you can look here and you can say, wait a minute, and so there's some place in that genome right there that's controlling activity. Now that's fabulous, but that's actually 20-year-old genetics theory. Hasn't been quite that simple. There have been quite a few genes that have been suggested to be associated with this. This was a table I did several years ago in a review paper. Dopamine receptor 1 in the brain, this one called nascent helix, loop helix 2, glucose transporter 4. Some of you may recognize that, GLUT4 in the muscle, myostatin, some of you have heard of that, aromatase, and a variety of others. There's very limited support for any of these down here. But so there have been some suggestion about the genes that are controlling physical activity. But I'm going to tell you that we don't know for sure yet. We don't know for sure, okay? As we were getting to this, we thought we were missing something. Something wasn't working here. And so about 2010, I got very dissatisfied with quantitative genetics, with the approach that we were taking that my colleagues were telling me. I was working with several quantitative geneticists, and their assumption was, and most of the assumption that you'll see in exercise genetics is that you have some difference in the DNA, in the genome, and some variation in the genome is actually directly controlling your end characteristic, whether you're a couch potato or a frantic banana. And what the problem is so many people forget is there's a lot of steps between here and down here. So many of you, I know this may give you nightmares. We have the transcription, we transcribe it into mRNA. We have the translation where we transcribe it into protein. You have post-translation modifications. You have this new thing that really happened in 2006. Actually, a person won a Nobel Prize for describing this in 2006, some RNA interference mechanisms that prevents this from being translated into protein. So you got all this stuff going on here. And so I began to ask in 2010, I said, wait a minute, we got all this stuff going on here. This assumes if you have a variation here, you assume that all this stuff doesn't change till you get down here. The question I had, well, and what we found when we started looking at the literature, that it's just because something transcribed, just because you have a transcription, doesn't mean it translates into protein. And there are all these studies that have shown really, really clearly that just because you have gene expression doesn't mean that you have a protein. And if you don't have a protein, nothing works. Nothing happens. And so we started to ask the question, let's look at the end protein and work backwards. So let's look down here at the protein that happens here and work backwards up that line. And I'm not going to bore you too much with this, so hang with me. So we tried this. We're the only ones to have published anything with the proteome of activity. This is Dr. David Ferguson, who's now assistant professor at Michigan State. He came with me when I moved here in 2010 from Charlotte. And David graduated in 2012, I think, 2013 one. Anyway, David, for his dissertation, he did the proteome in both the brain and in the muscle of these mice, of the high and low active mice. And what he found was that there are both issues, or there are both genes or proteins in the periphery, in the muscle, and in the brain that control physical activity. I'm going to blow through these, because I'm going to get to this piece right here. So if you take our high protein animals, they got genes like creatine kinase B, succinyl-CoA ligase, annexin A6, calsequestrin 1. Our low active animals, they have a different signature. And so what David showed, the take-home message here, low active animals just don't have less protein of a certain protein than the high active animals do. They're all the same. They have different protein signatures. There are different mechanisms that are the activity in the low active mice and the high active mice. So if you have a predisposition to be low active, that doesn't mean that you're a defective high active individual. That means that you've got a whole nother set of genes that are working versus someone who's high active. Okay? So it's a different mechanism for both of these. David, actually, we figured out how to turn these genes off. We can turn these genes off in mice now, and I'm going to show you just one example of that. If my clicker will work, back up. Come on, where's the... Okay, well, that one didn't work. We're going to quit. So just take my word for it. David did actually 2 publications where we actually turned off those genes. We actually were so concerned about this technology, we actually talked to the USOC, because this is a technology that you could use in humans to turn off genes. And if you were to turn off things like myostatin, some of you are aware enough to know what myostatin does, you could actually cause huge increases in muscle mass, and it would be untraceable. So, because this stuff we were using actually degrades, and you wind up peeing it out, and there's no trace of it. And so we talked to the USOC about how to combat this if it happened, blah, blah, blah. So anyway, but it was very effective at working. Now, there's one factor in particular that is very, very effective at controlling activity. And I'm going to blow through this really quick. This is a guy named Kurt Richter. He was at Johns Hopkins, and in the 1920s, he published a bunch of papers that said spontaneous activity is dependent on ovarian function. So he said, before they even knew what sex hormones were, he said, wait a minute, something is happening that controls physical activity based on, in that time, female reproductive cycles. And this is one of my former doctoral students. I'm going to blow through this really quickly. What he showed is if you took males and you took females, and you actually took out their sex organs, if you did this, you notice that in both cases, with the males, you decrease physical activity on a daily basis by 90%. In females, you decrease it by 31%, maybe not 60%. If you gave them testosterone or you gave them estrogen, it brought it back. This, of all the things we've ever done, causes the biggest change in physical activity. You take away the sex hormones, physical activity goes away. If any of you were raised on a farm, you know that's why you do that with cows in particular. You castrate cows because it calms them down. Okay? So what so far? So we know that genetics is a controller of physical activity. Genomic approaches have resulted in verified controlling genes, some candidates, ambiguous results. Proteins have given us some candidate proteins. We think 2 proteins in particular are involved. sex hormones also play a role. That's to set up this next piece. And this question. So here's your next question. Any questions? Like, how did you get your hair to stand up or something like that? So any of you have any questions at this point? Raise your hand. That's right. Let's just go do the old-fashioned way. Yes, sir.
So is there as much variability in human beings as there are in those two types of mice you showed us as far as physical activities?
Yes. Yeah. Some of the big studies that have been done, especially in the Europeans, have shown that the variability in humans, the highest heritability rate is about 90%, 92%, was published by a group named, author was Josen, and the lowest is 20%. So there's a big range there. Some folks that have actually looked at that across the different countries, the Scandinavian countries have the highest heritability. There's a lot of things that can influence that. More recently, we're finding age of the subject really makes a big effect. Humans, up till the age of 18, it's really environmental, especially the environment they grow up in. Once they turn 18 and they're on their own, from 18 to about 50, genetics really plays a large role in physical activity. After that, if they get old, and that genetics starts to fall off. So there's a lot of things into that category. Yes, ma'am.
Is there a way in which you can actually turn on the physical activity? Like, because I feel like me personally, I was a lazy child, and now I'm the opposite.
It's a good question. I'm going to show you some data that we can think maybe the opposite way. How do we take someone how do we take someone that's high active and make them low active, and perhaps the opposite works? We've been asking for money to do the opposite. We've been working on that question. So, yes, ma'am.
Mine was kind of similar to hers. So, did training have any influence on the
The data that's been out there has not shown physical training has had any influence on daily physical activity. Actually, there were some studies done in the 50s where they took schoolboys, that's what they called them, in England, and they ran the heck out of them every day, and they actually found out they were less active. So that's led to a lot of theories, but there has been no direct anybody approaching that. Yes, sir.
Dr. Lightfoot, that when you breed high active and low active mice,
Yep.
crossbreed them,
Yep.
the traits are passed on to the grandchildren.
Yep.
Is that because of an epigenetic effect? And if so, what's the effect on the first generation after?
The first generation after are also high active, and so the high activity seems to breed through. As a matter of fact, we're the only ones that have published that kind of data. And actually, we were very excited when we found this. We did this in a cohort of about 400 animals, and so it was a very strong indicator that there was only 1 or 2 genes that were involved. Since then, I think it's probably more than that. Epigenetic factors, yes. I'm going to talk just a few minutes about a couple potential epigenetic factors. There's also epistasis as well. So we have genes on one chromosome that are affecting genes on another chromosome also. Yeah. Other questions before we go on? Nope. I got 8 minutes, right, Mike? Good. So
You got their attention. So keep it going.
here's the biggest thing. I want you to think about this for a second. If there is a biological mechanism that controls how active someone is, if it's not necessarily voluntary, then what about, are there things that we're exposed to on a daily basis, whether it be training, whether it be something else that controls how active we are? I'm going to give you 2 of them to think about, and one of them, they may be epigenetic. Danny, I don't know. We're still working on some of that. So let's talk about a common environment. There's something called phthalates, and phthalates are toxicants that are common in our environments. They're in carpets, they're in sheetrock, they're in building materials. More importantly, and I don't know if I have this in flooring, in particular they're in personal care products, especially in makeup that females use. These phthalates are anti-adrenergic, they're weak anti-estrogenic. So people have thought, well, they're not anti-estrogenic, so we're not worried about them for females. Okay? Human embryos are exposed to multiple phthalates. They pose a large issue in utero. And so we asked ourselves, does this affect physical activity? If sex hormones are involved in regulating physical activity and phthalates control or affecting these hormones, does this affect the offspring? And so we looked at one called benzoyl phthalate, which is everywhere, BBP. This is Dr. Emily Schmidt. She was one of our doctoral students here. She's now doing a postdoc over actually in environmental toxicology. And the short answer is yes. She took a group of mice. She exposed the mothers She got them pregnant. She exposed the mothers to BBP, relevant dose, and they had the offspring. She then measured those offspring, followed them for 20 weeks. And what you can see here, so this is the amount of distance they run on a daily basis. This is males up here, females down here. These are the BBP animals. These are the control animals up here. And you can see it's not a huge difference, but it's in males it's about a 10% difference over that 20 weeks. And over females, it's about 15%. What the data we don't have yet, she's still working on it. Actually, she's putting a manuscript together. She showed in the female animals that were exposed to this, they have huge rates of cancer, mammary gland cancer. And mice don't get breast cancer. They don't have breasts. They have mammary glands. They don't get mammary gland cancer. And the fact, when we did this and when she showed this at a cancer meeting, we had people going, what? These mice don't get cancer. How do they have cancer? It's like, all we did was expose them to BBP. That was all. Anyway, so this told us that certainly there may be some environmental exposure that's affecting things. And if this, Danny, if this is passed on to grandchildren, I don't know. So we don't know if it works epigenetically or not. We actually have an NIH proposal in looking at that. Let me give you one other factor, and then I'm going to wrap this up. And it's feeding. There is an activity continuum. If you, we know that if you actually restrict calories to humans and to animals, 8 to 12%, you actually will drive their activity. You will increase their activity. And again, this has been shown in humans and animals. Regina Casper has actually done a lot of work with anorexic patients, and she suggests that actually the decrease in caloric intake in anorexic patient is what drives their activity, their high levels of activity. We have asked the other question, does it go the other way? If we increase how many calories they eat, do they become couch potatoes? And so there's been a couple of human studies that have suggested this may be the case. A friend of ours down in San Antonio that worked with primates, they have anecdotally seen this in the baboons they have down there, that when they give them sugar water as opposed to water with fructose in it, these animals stop climbing. They actually lay around the water trough. So we thought, well, let's try this. Let's see what's happening. And so former doctoral student, Dr. Heather Vellers, who's she's doing a postdoc right now at the National Institutes of Environmental Health Sciences. She took and she gave mice high-fat, high-sugar diet for 10 weeks. And this is what she found with activity. Now, these are animals that we bred, so there's no breeding issue here. The offspring came out, she randomly assigned a mouse to a high-fat, high-sugar or control diet. And if we just look at the distance, this is how far they run on a daily basis. These are the males, and here's the females. The red are the controls. The blue are high-fat, high-sugar. You can see the animals on the high-fat, high-sugar diet actually were about 62% less active. Duration, how long they ran on a daily basis, you can see there's a decrease here. Speed, they ran, they exercised at about the same intensity. They just didn't go as long. This is not the first time we've done this. We've actually seen this, I believe, in 3 other studies, right, Jorge? We had a big study with the Department of Defense where we first saw this as well. So this is huge. This means that if you overfeed an organism, that they are less likely to be active. So think about our children now. They are certainly exposed to environmental toxicants. They are certainly exposed to overfeeding. We wonder, well, these kids aren't active because they just don't want to be active? Or is there something else interfering with it? Maybe something else interfering with it. So there's some exciting things happening in genetics. Genetics has changed so much in the last 20 years. What we know and what we think we know, things like miRNA, epigenetics, these are all changing. Gene editing is coming to reality, vivo morpholinos, which we have used, this thing called CRISPR. If you haven't heard of CRISPR, you should look at CRISPR, because it's scary, actually. Because it's cheap, anyway, we can get a CRISPR cassette for $75 and we can knock out any gene we want. The morpholinos cost us about $4,000 to do one gene. CRISPR is about $75. How it all applies to exercise and performance, we don't know. We don't know. Why should you care? Because there's more and more emphasis on determining genetic underpinnings of performance. There's a lot of people out there that are saying we will test your genome and we will tell you how you will perform, what diet you should eat. I saw one for the microbiome the other day. What food you should eat so you have the best microbiome. 23andme.com and others. There's not good evidence for any of this. A really quick story. Hey, athletics came to us a couple years ago and said, should we do genetic testing on our athletes? Other universities are doing. One up the road, a place called Waco, is doing this because they think it will make them better. The answer was no, because no one knows what they're doing. It's crap right now. There are companies that are making money, but they're not giving you anything important. So eventually we will figure this out, though, and it'll be something else that you can use for exercise prescription. Some take-home messages. Genetic biology plays a large role in determining daily physical activity. Exact gene or mechanisms are still unclear. Good candidate proteins. Genetic biological influence implies that the mechanism can be altered. So we know there are some direct environmental influences that alter physical activity. And let me tell you a couple of professional ones to take home with you. If you're staying in this field, find your passion and pursue it. No matter what anybody tells you. I said I was interested in space physiology, and people said, you're nuts. Yeah, right. I am nuts, but yeah. Surround yourself with a supportive network. Don't be shy. Talk to people. Ask them. Share with them what you're interested in. You'll be surprised. They want to help you figure that out, where you're going. These were some of my buddies at Tennessee who I've still stayed in contact with. I'm going to skip that. Stay aware. Pay attention to what's going on in the field. And I always ask people to be professionally graceful. Help people go through the business like you're going through it. With that, I think we have any last questions, and I'll just take them in a traditional manner. Any last questions before we tell you guys goodbye?
How are you exposing those mice to the UV?
We were doing what they call Worrell Gavage, so we were basically squirting it down their mouth. So, which is actually supposedly the most physiologically realistic way to do it. Since we get primarily BBP through, we either absorb it through our skin or we eat it. It's actually in foodstuffs. We breathe it, so it usually goes in our respiratory tracts and in our stomach, a variety of other ways. Yes, sir.
I think it's really interesting because there's a big push in health right now to find causes beyond genetics for uterine cancer.
Yep.
And one of the main things that people are honing in on is feminine hygiene products because they're not regulated by the FDA.
That's correct.
So I think that's
If you were pregnant, ladies, when you get pregnant, you should stop putting makeup on. Or you should really make sure BBP and some of the other toxicants are not in your makeup. So, and more because they are, which is really scary. Guys, when your spouse gets pregnant, you should be talking about that. Pull in all the literature, especially early on in pregnancy when these things are developing. I mean, we were hitting our mothers early on in their pregnancy with this stuff when all that formation was happening.
We thank Dr. Light. You got questions yet?
How can we enjoy sports for our lives since we know more about genetics? Because maybe our path is like destiny for we know about genetics. I mean, everything is known for your gene.
Yeah, well, not everything's quite known. That's the more we know, the less we understand. And the one thing I want you to take away from this study is this story today is not that we are absolutely predestined to be inactive or predestined to be active. It's just a predisposition. The numbers are such that we can overcome that. And it's the same way with endurance, it's the same way with trainability. All these things are predispositions and not predestinations. So, we can still overcome our genetic influence.
I know there's another class trying to get in here.
Yep.
Dr. Lightfoot, come in.
Thank you. And so I was going to award these. I'm going to give these to Dr. Greenwood, and you can choose. Here, I'll
That's
choose.
too much pressure.
You know what? Because we had people that gave us the questions first off the bat. So this young lady here, and give this to this gentleman in behind, even though he walked in late.
He's not in this class. You get a present for being late.
There you go.
Busting in